Hey, welcome everybody to a new Global IMMUNOTALK. I'm Carla Rothlin, and together with my friend and colleague Elina Soniga, we have organized this series of talks. And just briefly to remind you that our main goals are to make immunology accessible to as many of us across the world in an egalitarian manner, and to increase the opportunities for this scientific learning without traveling. We cannot stop thanking our fabulous speakers that have accepted our invitation. We have today Jen Gommerman, that is going to be introduced by my friend Elina. And just to remind you, we'll have Katrin Meyer-Barber next Wednesday joining us in this Global IMMUNOTALK series. So with this, I'd like to ask Elina if she can introduce our speaker of today, Jen. Thank you.
Perfect. Thank you so much, Carly. It is my real pleasure to introduce to you Jen Gommerman. She is professor and associate chair of the graduate studies at the Department of Immunology at the University of Toronto. Jen is native from Canada, is our first Canadian speaker, and she was born in Winnipeg, Manitoba, where I understand the weather can be as cold as Siberia. So it would be interesting to know how that shaped your sense of community, Jen. She has lived most of her life in Toronto, where she received her bachelor science degree and her PhD degree in immunology at the University of Toronto. She then went on to perform a postdoctoral training at Harvard Medical School, studying the complement pathway, and afterward she joined Biogen as a staff scientist. That was in 2000. During her tenure at Biogen, she became interested in B cells, multiple sclerosis, and the TNF superfamily of molecules. After 3 years in industry, Jen returned to academia as an assistant professor in immunology at the University of Toronto in 2003, and in 2015 was promoted to full professor. Jen's basic research continued in academia to focus on how members of the TNF superfamily of molecules regulate immunity and autoimmunity. Her team has uncovered novel gut-brain axes that regulate neuroinflammation. With respect to translational work, Jens has been examining the role of B lymphocytes in multiple sclerosis patients, and she is the lead PI on a study examining the effects of global migration on susceptibility to autoimmune diseases. More recently, she has been studying the antibody responses to SARS coronavirus 2 in saliva samples from patients with COVID-19, and I think we will learn more about this recent work during her talk. So Jen's amazing discoveries have been published in top-tier journals, including Cell, Nature, Science Immunology, among others. And she has received many awards for her discoveries, such as the Canadian Society of Immunology Investigator Award, and has also been honored for her mentorship trajectory, receiving in 2016 the University of Toronto Faculty of Medicine Mentorship Award. So Jen, it's a pleasure to host you. Thank you so much for making time to prepare the talk and to be here presenting today.
Thanks for the invitation. It's great to be here. And thank you for setting up this seminar series. I think it's been a really great way to maintain a sense of community in a very, very strange time. Thank you.
Thank you, Jen. We actually feel super lucky to have been able to do this. So another fact about Jen that I haven't yet shared with you is that she is also the director of the University of Toronto graduate program in immunology that has over 100 students. And so as such, she serves as advisor and role model for students. And so with that in mind, we would like to ask you, Jen, what is the number one thing that the students come to you about?
Okay, well, first of all, thank you for asking. I know you do this question period at the beginning of each talk, and I think it's a great way to begin the science with a little bit of humanity. And so I guess the top question or the top concern that students bring to my office is, I think, an overwhelming sense of imposter syndrome. And I think that this is something I've noticed a lot in students, particularly lately, and even more so during the pandemic. And I think it's important to acknowledge that we work in a fairly rarefied environment. We're taught to be very critical, and we're surrounded by really smart people. And it's important to kind of get some perspective. And so what I always tell students is to, first of all, that they're very smart and that they can do this, and that it's a major shame when someone leaves science because they don't think that they have the potential to succeed. And secondly, I think it's also important to acknowledge that that faculty members and really people from all along this hierarchy, for better or for worse, it is a hierarchy, experience this feeling, especially PIs, and that we're not immune to it. And so the way I handle this is to often share some of my own experiences with students where I certainly felt like I was faking it and didn't belong in the environment I was in, but really I was just fine. So this is a big issue, and I would, if there are trainees listening, I would encourage you to not ignore that. We don't ignore it when our car makes a funny noise, and we take it in to be repaired. And likewise, if you're feeling really overwhelmed and anxious, I really recommend students to take care of that and to take care of themselves so that they can be the best scientists that they're really meant to be.
That's beautiful, Jen. Thank you so much. I'm sure it's very helpful for students, postdocs, and PI. And I echo your words. I think as PI, we are not at all immune to the imposter syndrome. So thank you so much for sharing your thoughts about it. Yeah. Okay. So we cannot wait for your talk. So if you could share your screen. Let's see if it works. Okay.
Can you see it?
Yes, we can see it perfectly fine.
Perfect. And this is the first slide you can see?
Yes.
Great. Okay, well, I'm going to take it away then. So in addition to thanking Alina and Carla, I also want to thank you, the audience. And I think it's very important to acknowledge that, that it's hard to take an hour out of your day, especially these days when we're juggling so many different things that we ordinarily wouldn't be putting together our personal and professional lives in the way we have been. And many of us are having to care for children or care for elderly family members and take Zoom calls and color our own hair and do all these things. And I gotta say, the multitasking is driving me crazy, and I'm sure it's driving you crazy too. So thank you for joining me. Now, when I think of multitasking, I think of plasma cells, and that's probably not what you think of, but it's what I think of. And you probably think plasma cells have really just one job, and that is to make antibodies. And hopefully what I'll convince you of today is that plasma cells actually can do other things besides making antibodies. And in particular, I'm going to focus on plasma cells that make the antibody IgA. The gut is really the largest reservoir of IgA producing cells. And in and of itself, this is actually a very, very significant reservoir of immune cells. And we're going to basically, I'm going to take you through 3 questions today. The first is, what are some early life signals that can impact IgA responses to virus? Is there a role for IgA plasma cells in neuroinflammation? And these two are both published data, and then I'm going to move to some unpublished data also on neuroinflammation and our recent work on IgA in SARS-CoV-2 infection. So this is a beautiful diagram that many of us use over and over again from this review. I've adapted it because I think it's a great way to illustrate how B cells are primed to become IgA producing cells within the gut-associated lymphoid tissues. So B cells are primed within what I like to call inductive sites in the Peyer's patches and in the mesenteric lymph nodes in response to commensal antigens or perhaps foreign antigens such as viruses, which I'll be talking about today. And within these environments, if there are cytokines that promote class switch to IgA, which is typically the case in the gut, then a B cell will switch to making IgA from IgM to IgA. And this requires an important enzyme called activation-induced cytidine deaminase. Within the mesenteric lymph node, this occurs in a germinal center environment that contains T follicular helper cells to make sure that there are the appropriate cytokines there, as well as CD40 ligand. And B cells that are primed within the germinal center and class switch to IgA begin this program where they become a plasma cell and they egress through the efferent lymphatics. Then they go through the thoracic duct to the blood, and they migrate all the way back to the gut into the lamina propria. And here they make IgA antibodies, which have the capacity to cross the epithelium due to the secretory chain. And these IgAs, which are dimers, can bind to the commensal microbiota and shield it away from the host, which is important for maintaining homeostasis. But it's also a great way to deal with infections within the gastrointestinal tract. We know that these plasma cells actually sit in the gut for very long periods of time. Some carbon labeling experiments in humans show that the most differentiated of these plasma cells that lack B220 and CD19 can live in the host lamina propria for decades. So we've been studying IgA plasma cells for many years, primarily prompted through early observations of defects in mice that lack lymphotoxin. So the lymphotoxin pathway, I first started studying this at Biogen with Jeff Browning, beginning in 2000 to 2003 while I was there. And it's a really fascinating pathway that's important both in utero as well as in the adult. This receptor, LT beta receptor, is a TNF superfamily member that's found on support cells, so we'll call them stromal cells, but also myeloid cells, including dendritic cells. And it's activated by two ligands, LT alpha beta and LIGHT, and these are predominantly expressed on activated lymphocytes. And if you lack these molecules in utero, you have defects in forming lymph node and Peyer's patches. And you can inhibit this pathway both in utero and in the adult with this fusion protein called LTBRIG, which consists of the extracellular domain of the lymphotoxin receptor with an Ig tail. Yang-Xing Fu and others have shown for a number of years that if you look at IgA levels in lymphotoxin deficient mice, there's virtually no IgA in the serum or in the fecal pellet. And it's still not entirely clear why this is the case. And there's a number of possibilities. One is that what Yang-Xing showed was that lymphotoxin beta receptor signaling on radioresistant cells, such as stromal cells, is important for setting up a milieu that's important to support IgA class switch and IgA plasma cell recruitment to the lamina propria. We don't think that this defect in IgA is due to an absence of organized lymphoid tissues, because if you get rid of mesenteric lymph nodes and Peyer's patches, you can still make an IgA response. So it could be that this stromal cell signaling might be playing a role. But we know also from beautiful work from Jason's sister's lab that signals within the Peyer's patches in dendritic cells that express LT beta receptor are also very important in the early stages of a germinal center response, promoting class switch to IgA. So this is obviously a very complicated pathway. And one of the common denominators to these studies is that they've looked at IgA responses, for the most part, to the commensal microbiota. But we know that as soon as you're born, you acquire commensal microbiota, and the IgA response follows very quickly thereafter. So this is an immune response that's occurring throughout your lifetime. So it's difficult to follow changes in this immune response. Moreover, from vivarium to vivarium, there can be major differences, of course, in the microbiota that in turn can impact the levels of IgA. In fact, Jeremiah Faith has recently shown that even strain level differences in the microbiota can profoundly change the levels of IgA in the gut. So we wanted to look at this question from a different angle, and that was to look at an antigen-specific IgA response in the gut. And we chose to look at this rotavirus model. This is a mouse-adapted version of the human pathogen that we got from Harry Greenberg's lab. And Sean Lay, a postdoc and a research associate later in my lab, has taken this on. And he developed a way to track B cells in vivo in response to rotavirus infection. And he uses a number of methods, including just looking at anti-rotavirus titers in the fecal pellet, as well as ELISpot. And actually, I'm going to use ELISpot as a readout quite a bit in this talk. So you administer the rotavirus orally, and then you can use it to look at a de novo immune response, which we start to pick up around day 7 post-infection. So the first question Shanley had was, where is the B cell response, B cell activation in response to rotavirus taking place in the host? We know that rotavirus comes through the Peyer's patch. It's been nicely shown by Brian Kelsall and others, but we didn't know where that B cell priming event was occurring. And to do this, Shanley took advantage of reporter mice that report on AID expression that we got from Rafael Casellas' lab. And what he did was he looked at the expression of AID as a marker of early B cell activation, because this is the enzyme that's needed for the B cell receptor to undergo secondary diversification. And what he found was that in response to rotavirus, you see a bump in AID expression, which isn't 0 to begin with, because there is a constant immune response going on to the commensal microbiota. But you see an induction around day 7, around the time when we start to see antibodies made in response to rotavirus infection. But he only sees this in the mesenteric lymph node, and he does not see this in the Peyer's patch or in the small intestinal lamina propria. And this bump in AID is also coincident with the appearance of rotavirus-specific IgA secreting cells that he measures by ELISpot. And these ASCs are seen in the mesenteric lymph node as early as day 8 post-infection. And at that time point, they're not seen in the Peyer's patch. You're already starting to see them in the lamina propria, however, and we think that this is rapid migration of generated plasma cells out of the lymph node and into the lamina. So the next thing he asked was, is there a role for the lymphotoxin pathway? And if so, when does this pathway play a role? Because we know it's important both during development as well as in the adult. And what, I'm not going to show you the data, but Shanley did inhibit the pathway in a variety of ways in the adult and did not see a defect in the rotavirus response. So you can inhibit this pathway as much as you want in an adult animal, but the rotavirus response is fine. So he had a look at whether the pathway was relevant if he just inhibited the pathway in utero. And to do this, he took advantage of the LTBR-Ig reagent that I mentioned before, which many years ago, Paul Rennert and colleagues had shown because of the Ig domain, it can cross the placenta in a pregnant dam and impact and inhibit the lymphotoxin pathway in the developing fetus. And so he gave this fusion protein at E14 and E17 of development. The mesenteric lymph nodes are not impacted by this particular kinetics of treatment. As you can see here, at least grossly anatomically, they look pretty normal. And the mice that the pups that were derived from the LTBR-Ig mice, he mixed them with pups that were coming from control-treated dams to normalize the microbiota. And waited for up to 8 to 12 weeks until the mice had reached adulthood, and then infected them with rotavirus. So just keep that in mind, that all of these experiments are looking at an inhibition event that occurs in utero, but all our readouts in terms of the infection are in the adult. He also made bone marrow chimeras, which likewise prevents signaling in utero, but restores signaling in the adult. But the advantage here is that we can now say that the impact of the pathway, if there is one, is through the LT alpha beta ligand as opposed to light. So this was a sort of complementary approach that he used. And what he found was that using these 2 injections of the inhibitor, if we looked at the rotavirus response, there was a defect in the early stages of the immune response in the lymph node. This is now tabulating the number of rotavirus-specific IgA plasma cells by ELISpot. This carried out to day 49 in the lamina propria, and the titers of anti-rotavirus IgA antibodies were significantly decreased. He saw the same thing in the chimeras, therefore implicating LT alpha beta, LT beta receptor signaling in this process. So here we have a signal that's coming early in life, in utero, that we've interrupted, and then the drug is long gone, and we've waited until adulthood, and there's now an impact on the immune response later in life. So obviously we wanted to know what was going on and how this worked. And we focused on the mesenteric lymph node because that was where we were seeing the priming. And We looked at a whole bunch of different steps of the germinal center response. We looked at the accumulation of T follicular helper cells, which are, as I mentioned, are important for sustaining and promoting a germinal center response. And we did not see a difference in these cells in the lymphotoxin inhibited animals. We also looked at the expression of AID throughout the immune response, and it was found to be normal in the lymphotoxin inhibited animals. We looked at different discrete steps in the germinal center response, and these were all found to be normal, if anything slightly enhanced in the lymphotoxin inhibited animals. And then we looked at the accumulation of plasma cells, and to do this we used this BLIMP reporter animal, which I'll come back to later, but BLIMP is a transcription factor that's very important for the differentiation of germinal center B cells into plasma cells, and it's required to maintain the plasma cell phenotype. And using this BLIMP YFP reporter, we found lots of BLIMP positive cells generated in response to rotavirus, but they appeared to be normal in numbers, if anything, slightly increased. So all these things were normal, and actually that was kind of comforting because we were inhibiting this pathway in utero. We know this is a time point when there can be lots of developmental things going on, and yet all of these steps that make that form discrete parts of the IgA response in the mesenteric lymph node appeared to be fine. But what was not okay was class switch itself. So what Shanley had noticed was that actually, if he looked at ELISpots for IgM, he found that they, in the lymphotoxin inhibited animals that got this inhibitor in utero, that the number of ELISpots that were rotavirus specific but had not class switched were actually normal, but it was the IgA class switched ones that were reduced. So when you express this as a ratio, you get this big reduction in lymphotoxin inhibited animals. And the same was true if he looked for germline transcripts. So what he did was he sorted out on cells that were actively expressing AID using those reporters, and then he measured the amount of germline transcript, for example, the C alpha, and this was found to be significantly less in the context of mice who did not have lymphotoxin signaling in utero. So it seems that if you inhibit this pathway early in life, there's an impact later in life in terms of the ability of these B cells to class switch to IgA. And I should add, I don't have time to show the data, but if you look at the B cells themselves that are undergoing class switch, they also appear to have a defect in their ability to upregulate CCR9 and alpha-4 beta-7. These are markers that are required, these are homing receptors that are required for the IgA positive B cells to home back to the lamina propria, like I showed you back in the beginning in that beautiful review figure. So this could be a sort of 1-2 punch where you have a defect in class switch, but then you also can't seem to move these B cells down to the lamina propria after they've been primed. Now, we don't think this is because there's a problem with the B cells themselves, because if we take them out of the animal and ask them to class switch in vitro by adding switch factors, etc., that's totally normal. So these mice that lack lymphotoxin signaling in utero have B cells that can switch absolutely normally. And I mentioned that lymphotoxin beta receptor is expressed on stromal cells in the beginning of my talk, and it's actually very important for maintaining stromal cell identity. And so we wondered whether the stromal cell environment of the mesenteric lymph node might be altered when you inhibit this pathway in utero. And so we had a look at that in the mesenteric lymph node during rotavirus infection, and what we did was we sorted out all of the cells that were not stromal cells. We got rid of all the hematopoietic cells, any plasma cells that expressed BLIMP, we got rid of them. And we focused on the CD45 negative cells, and we did this classic stain that was originally described by Shannon Turley and colleagues to divide up different types of stroma within the lymph node. And we focused on these cells that expressed GP38, or otherwise known as potoplanin, and did not express the CD31 endothelial marker, because these are putative fibroblastic reticular cells. Now, we fully acknowledge that these cells are quite, no doubt, very heterogeneous, and the more people are doing single cell RNA sequencing, the more subsets of FRCs are being discovered. But we wanted to look at this population in bulk, and what we found was that during the rotavirus immune response, there were far fewer of these cells in the mice that received this inhibitor in utero. And if we did bulk gene expression analysis, there was quite a few differences with the remaining FRCs of the lymphotoxin inhibited animals compared to the controls. And some of the gene expression differences that we thought were particularly interesting were these here, which are all involved in the IgA pathway. So for example, we have BAFF and APRIL that are decreased in the mice that got the inhibitor in utero. IL-6 is decreased. It's an important plasma cell survival factor. MMPs 2 and 9 are decreased, and these are important for cleaving TGF-beta into its active form, which is a very important IgA switch factor. And then also members of the retinoic acid pathway that we know are important for imprinting gut homing potential were also reduced. So this kind of makes sense, and we concluded that these changes in stromal cells may impact the IgA immune response. And in fact, if we delete LT beta receptor only in FRCs using a CCL19-driven Cree, and I don't have time to show you the data that we got from Burkhard Ludwig's lab, we see basically the same phenotype in terms of the rotavirus response. So this was strong evidence that the pathway in utero was programming stromal cells in order to foster IgA class switch events within the mesenteric lymph node in response to rotavirus infection. And so that's just summarized here in the first part of my talk. So the next thing we wanted to do was to have a look at whether these cells might impact other tissues outside of the gut. And I'm going to walk you through why we took this leap. And the way I'm going to walk you through this is by talking to you a little bit about a clinical story that was in the literature and that really got our attention. And this clinical story is about a disease that we study a lot in the lab, multiple sclerosis. For those of you who are unfamiliar with the disease, typically it's diagnosed as a relapsing remitting disease that is marked by bouts of clinical and cognitive disability that coincide with inflammation in the white matter of the brain and the spinal cord. This is due to the migration of immune cells through a breached blood-brain barrier into the white matter, where they cause pathology, in particular demyelination of the myelin sheath that protects axons. And as the disease progresses, these inflammatory episodes actually become less common, and yet the disability persists, if anything gets worse, in the secondary phase of this disease. And I'll come back to that in a bit and explain what we think might be going on here. Okay, so relapsing remitting MS can be treated reasonably well with immunomodulators, which makes sense because it's really the peripheral immune system that goes into the brain. And so drugs like siponimod that prevent lymphocyte migration, or Tysabri that block entry into the brain, are very effective at treating MS. And for many years, T cells were thought to be the main movers and shakers of this disease. We know that differences in HLA can be major risk factors for the disease. And if you prime T cells now in an animal I'm talking about, and you prime them to myelin, and then you transfer them into a naive animal, those T cells are on their own sufficient to induce an MS-like disease that we call experimental autoimmune encephalomyelitis, or EAE. So T cells are kind of the stars of the disease. And B cells have been a little bit on the sideline. We haven't really understood their role in this disease as much as what we understand about T cells and the cytokines they make, which in particular are interferon gamma, IL-17, GM-CSF, etc. But yet we know that there are B cells in the MS CNS. So if you look in the cerebral spinal fluid of MS patients, you see these unusual bands that correspond to antibodies. And this is abnormal. You don't see this in a healthy person, and used to be a marker, a diagnostic marker for MS, and is still used in combination with MRI in many places to diagnose the disease. So this is really a hallmark of MS. B cells are in the MS CNS, and I'll come back to this observation, but they typically are in these brain adjacent regions, for example, in the leptomeninges. And so they're there. Whether they play a role in the disease was debated, but I think a major key experiment was done by Hauser and colleagues at UCSF, where they showed that if you deplete B cells with an anti-CD20 antibody called rituximab, which until then had been used to treat lymphoma, that this has a remarkable effect on MS. And MS patients that get anti-CD20 antibodies, their burden of new lesions goes down to almost zero within 12 weeks. And this has really become probably one of our best treatments for MS patients. Now, interestingly, it was thought that maybe this drug had succeeded because it had had an impact on these B cells that are making antibodies in the CSF. The precursors for plasma cells or plasmablasts had been deleted with this antibody or depleted with this antibody, and therefore this pathology would have been attenuated. But actually that proved to not be the case. And if you look at MS patients that got anti-CD20 and had a positive response to the therapy, you can still see antibodies in their cerebrospinal fluid. So this effectively dissociates the antibody producing part of B cell biology from the efficacy of anti-CD20 treatment. So this was maybe not so surprising, because if you actually look at the expression of CD20 on B lineage cells, by the time you reach the plasmablast stage, after B cells become activated, CD20 starts to be downregulated and is pretty much gone on the surface of plasma cells. So these cells are not targeted by anti-CD20, and because they're so long-lived, that could potentially explain why those antibodies persisted. So the thinking was, maybe we should try and get rid of the antibody-producing cells in MS in addition to the garden variety B cells, because there are forms of MS that are resistant to anti-CD20 treatment. So maybe if we do that, there'll be a better efficacy. And to do this, a fusion protein called TACI-Ig was tried. So TACI is also a TNF superfamily member. It binds to these two cytokines, BAFF and APRIL. And these cytokines are very important for B cell survival, but also plasma cell survival. So by neutralizing BAFF and APRIL, you're virtually starving B cells as well as plasma cells, which will be reduced. And that's indeed the case. If you look at rheumatoid arthritis patients that got atacicept, their level of antibodies in the circulation dropped dramatically. And this is not seen with anti-CD20. So then the question was, is this going to be useful for MS patients? And so it was tried in 2 different clinical trials. And as you can see here, there was actually a very surprising result, which was that compared to the placebo, MS patients that got atacicept actually did worse when you looked at their clinical disability. And this was actually a dose-dependent phenomenon that was so obvious that they had to halt the trial and unblind it. And sure enough, it was a drug-dependent effect. In a second trial in optic neuritis patients, patients that got the atacicept actually had a higher conversion rate to MS from optic neuritis, which can be a precursor to the disease. So this was a bad news story for MS patients. Obviously, atacicept was not going to work. But between these observations and the anti-CD20 observations I told you about, we decided to test a new hypothesis. And our new hypothesis was that plasma cells may play a protective role during MS. Perhaps they're involved in suppressing inflammation. And we decided to test this hypothesis in the context of the animal model for MS called EAE. So this is work that was done by Dr. Olga Royas in the lab, who was a postdoc and then a research associate, and has now accepted a faculty position at the Krembil Brain Institute at the University of Toronto. Now, as I mentioned, plasma cells are the sort of the terminal end of B cell differentiation, and they're actually kind of hard to find in the blood, and that's because they need those survival factors, BAFF and APRIL, for example, to persist, often for decades in humans. And this, there are niches within the bone marrow that are rich in these survival factors. And so that's why we don't see them in the blood. And if they're generated in the gut-associated lymphoid tissue, they would home to the lamina propria, like I showed you for rotavirus. So these plasma cells, of course, we know are primarily known for making antibodies. But emerging literature has shown that they can also make other molecules, such as cytokines and immunomodulatory factors. And in particular, Simon Filatreau and others have shown that plasma cells are an important source of IL-10, which is an anti-inflammatory cytokine. And Simon also showed that plasma cells can actually express checkpoint inhibitors to attenuate the immune response. In addition to plasma cells being immunosuppressive, there's evidence that gut-derived plasma cells, in particular those that make IgA, are likewise immunosuppressive. And there are 2 papers from Michael Karen's group that looked at this in the context of cancer, both in mice and humans. So we wondered whether some of the exacerbations of MS that occurred during atacicept treatment could be due to a reduction in plasma cells and maybe even reduction in IgA-producing plasma cells. So to test this in EAE, Olga used these reporter mice that I told you about, BLIMP1. She used other reporters as well, but we'll just focus on BLIMP1 today, to map these plasma cells, which are hard to find because they downregulate so many of their markers and they can be difficult to extract from tissues. So she really worked hard to do this and looked for these cells in the brain and spinal cord during EAE. So just a sidebar on EAE, for those of you not familiar with the model, it can be executed in susceptible strains such as Black 6 or SJL. You do this by immunizing mice with peptide that is part of the myelin sheath, so it could be MOG or PLP in adjuvant. And then this develops a chronic disease or a relapsing remitting disease depending on the strain. But you can also take immune cells out of these immunized animals before they show symptoms, culture them, and skew them towards cytokine producing cells, particularly Th17 are known to be quite pathogenic for EAE, and then transfer them into naive mice and monitor disease following transfer. And we do both of these things in the lab. And so using the Black 6 EAE model, Olga showed that there is an accumulation of plasma cells in the brain and the spinal cord, but only once the mice start showing symptoms of EAE. Just immunizing them is not enough. They actually have to show the symptoms in order to find themselves in the brain and spinal cord. And when she looked at these plasma cells a little more carefully using FACS and ELISpot and other techniques, what she found was that they were often class switched, but a number of them actually had class switched to IgA. And this was surprising because, as I told you in the first part of my talk, the gut is really the major reservoir for IgA producing cells. And so this raised a whole bunch of questions. Are these cells actually impacting the clinical course of EAE? Where do they come from? And if they are impacting EAE, how are they doing this? So Olga first asked this question in the context of all plasma cells, and she basically deleted them by using a floxed model for BLIMP, crossed to two different CREs, CD19 CRE or AID CRE. She got the same answer in either case, and that was that if you remove plasma cells genetically, you actually have a higher burden of EAE disease. And this is consistent with other findings that were published actually prior to ours. So this is kind of like the atacicept finding, where if you get rid of plasma cells, as would have happened with atacicept, you actually have worse disease. And the next thing she wanted to do was ask, okay, are IgA plasma cells, which had been implicated in immunosuppression, playing a role in limiting EAE? And to do that, she took advantage of these plasma cell deficient mice and transferred plasma cells back into these animals, but she took them from the gut. She sorted them based on BLIMP expression. In a post-sort analysis, they were found to be almost entirely IgA positive, and compared them with gut resident B cells from the same animal. And only the plasma cells and not the B cells were able to attenuate disease in these plasma cell deficient EAE mice. So this told us that plasma cells derived from the gut could suppress EAE. This also implies that plasma cells might be migrating out of the gut, which is kind of a new idea. So we wanted to really take a step back and ask whether this happens in the steady state. And to do this, we went back to the rotavirus model that I told you about in the first part of my talk. We infected mice with rotavirus, and then in collaboration with Clint Robbins at the University Health Network, performed parabiosis. 60 days after the rotavirus it was long gone. The rotavirus is cleared in basically a week or two. And within 60 days, it's certainly gone. So that we put these animals together, an uninfected animal and a previously infected animal. And we asked if the rotavirus-specific plasma cells may find themselves in the peribiont, and that proved to be true. So here's the originally infected animal. These are rotavirus-specific IgA plasma cells, or ASCs, and this is the peribiont. And as you can see, there are some spots here. There's not as many as in the originally infected one, but there are certainly some in the gut and the bone marrow of this peribiont. So this tells us that, as my colleague David Allman likes to say, plasma cells, maybe they can go for a swim. And so that's maybe what happened here, is that they left the gut, went into the vasculature, and certainly we have evidence that they can enter a new animal. So then the question is this what's happening during EAE, and is that maybe one of the sources of these IgA producing cells in the EAE CNS? To do that, we infected mice with rotavirus, and then we induced EAE. And these are all the different controls that we did. And what Olga found was that if you look at these IgA rotavirus-specific IgA plasma cells by ELISpot. You don't see them in the CNS in the steady state. You don't see them if you immunize mice for EAE, but once the mice start getting sick, you see evidence of these cells within the CNS, both in the brain and then later in the spinal cord. And here's some spots. So yes, cells from the gut can migrate to the CNS. In particular, IgA plasma cells can do this. So since she'd shown that this was that these plasma cells were suppressive, we were very encouraged to see that they could actually land up in the CNS because this is a way of delivering an inhibitory signal to the CNS, which can be difficult to do with drugs because they don't really enter the CNS so effectively. We wondered whether we could actually harness these cells for therapy. And so how would we do this would be to potentially expand this pool of IgA plasma cells. Now I mentioned that these cells rely on BAFF for their survival. And actually APRIL, the other ligand for BCMA and TACI, is very important for IgA class switch. So inhibiting BAFF and APRIL seem to have this deleterious effect in MS. So we wondered whether having an excess of either of these two ligands would actually suppress neuroinflammation. So we actually had the perfect mouse to ask this question. We had been studying BAFF transgenic mice for a number of years. They were originally made at Biogen by Jeff Browning and Fabienne Mackay. And Jeff Browning and I later published that these mice don't have exceedingly high levels of immunoglobulin M or G, but depending on how much BAFF is in these animals, you can have really, really high levels of IgA. And this is a log scale. And not just IgA, but the IgA plasma cells themselves appear to be disseminated throughout the animal. So we thought, let's have a look at these mice and see how they respond to EAE. And as it happens, they were completely resistant to EAE. This is a mouse with 2 copies of the transgene versus 1 copy that has less BAFF, but still more than normal. And you can see that this is sort of a dose-dependent resistance to EAE. And it didn't matter what type of antigen we immunized the mice with, we got the same result. So this told us that BAFF transgenic mice were really resistant to EAE, but we didn't know what the mechanism was. And we thought that the first thing we would look at would be IL-10, because there was a precedent in the literature for plasma cells making IL-10 and IgA plasma cells making IL-10 in tumor environments. So we crossed the BAFF transgenic mice to the IL-10 deficient background. And as you can see here, mice with 1 copy of BAFF transgene are resistant to EAE, as I've shown you. But if these are on the IL-10 knockout background, we lose that resistance to EAE. And they basically look like wild-type mice. Moreover, if we take these BAFF transgenic mice that lack IL-10 and give back plasma cells from the gut, we can restore this resistance to EAE. But if we give back plasma cells from the gut that lack IL-10, we don't restore resistance. So this told us that an important source of IL-10 in BAFF transgenic mice that was conferring resistance was coming from gut plasma cells. And we showed this using a variety of methods, also looking at IL-10 reporter mice, and this held up. So the next question we wanted to know, I'm going to move now to some unpublished data, is whether this pathway might be important in the context of therapy. And so this is just summarizing what I just showed you, that IgA plasma cells play an immunosuppressive role in the CNS by making IL-10, and that this is really augmented by BAFF. So we wanted to know, what was the impact of these cells actually on brain pathology? And more importantly, for the therapeutic realm, whether this mechanism could be relevant to the efficacy that's seen, this incredible efficacy that's seen with anti-CD20 in MS patients, that's observed very quickly after they receive the drug. So as I mentioned, this disease starts as a relapsing form of MS and is characterized by these inflammatory lesions in the white matter that are really immune cell driven. But I hinted at the fact that this disease progresses and we don't really understand this phase of the disease so much. And as it turns out, the pathology in the progressive MS patient is actually quite a bit different. So as opposed to having these white matter lesions, there's still some white matter lesions at varying stages in progressive MS patients, but some progressive MS patients also show evidence of gray matter pathology. And in particular, they have these subpeel lesions, which are strips of gray matter that are close to the leptomeninges that are completely demyelinated. And we don't really understand what drives this pathology, but one potential smoking gun could be immune cells that have lodged themselves into the leptomeninges. So this is the envelope that surrounds the brain and the spinal cord. And within the leptomeninges, what's been observed in MS patients is these clusters of immune cells. And these clusters of immune cells, which are haven't been super well characterized. We know there are B cells, T cells, myeloid cells, some stroma, and that's about it. And that's certainly something we're looking into more. We know that these are adjacent to areas of the gray matter that are actually injured. So they're demyelinated, there are activated microglia in these places, and they look very unhealthy. And so what Richard Reynolds and Roberto Magliozzi and others have shown is that, and Claudio Lucinetti, have shown that these clusters of immune cells and the pathology associated with them seems to be bad for MS. It's a prognostically bad finding that if you retrospectively look at autopsy tissues and you look at the clinical course of disease, these patients that have these attributes typically have worse disease. So we wanted to model this in mice, and Natalia Picor, who was a graduate student in my lab and then went on to do postdoctoral work with Burkhard Ludwig in St. Gallen, Switzerland, and now has a group there. What she did was she did a variation on EAE in SJL mice where she primed immune cells in the animal, took them out, and then polarized them towards Th17 and adoptively transferred them into recipient animals. We knew that SJL mice actually have brain inflammation as a consequence of EAE, whereas black 6 mice, not so much. They're mostly spinal cord. So that's why we chose this model. And she does find immune cells within the brain, these clusters of immune cells, and they seem to be in these areas of the meninges that are within these sulci of the brain. And they're very much B cell rich. So these are full of B cells. There's T cells and dendritic cells there as well. Sometimes they're fairly organized, sometimes not. But they reliably happen in these locations. Valeria Ramaglia in my lab, a postdoctoral fellow now research associate, as well as Leslie Ward in the lab, went on to show that the pathology associated with these clusters of immune cells in EAE was closely associated with microglial cells. These are macrophage-like cells within the brain parenchyma that seem to sort of swarm up near where you find these clusters. And the membrane that separates the parenchyma from the meninges is called the glial limitins, and it is formed of astrocyte end feet, and this is completely trashed in the Th17 EAE in SJL mice, and the areas are quite demyelinated. So we were approached to test this model with respect to B-cell depletion, and we were skeptical because the black 6 models don't really respond to anti-CD20, nor does the active EAE in the SJL mice. Whether this model would was, we were doubtful. But Valeria tried giving a mouse-adapted anti-CD20 to pre-deplete B cells, and then she did the adoptive transfer and looked at the clinical progress. And actually, we found that anti-CD20 did impair EAE if you pre-depleted the B cells prior to adoptive transfer. So that was kind of neat and prompted us to have a look at what happened in the brain where these B cell follicles would normally be found. And what she found was that compared to control-treated animals that have clusters of B cells and T cells in the meninges, you obviously don't see B cells because they were already depleted, but you still maintain other cell types within the meninges. And we haven't looked at this super carefully. We're doing that now using a technology called imaging mass cytometry. But certainly there are immune cells there in the absence of B cells. And that's just quantified here. Now, what was really cool was that if she looked near these immune cell clusters, near the in the gray matter, she found this very severe demyelination, and we had seen that before. But if you look near the clusters that lack B cells in the anti-CD20 treated mice, the demyelination was much less. So the myelin was largely spared. and looked not as pristine as a naive animal, but pretty good. And so this suggested that the absence of B cells in these clusters actually is associated with less pathology. And we looked at all sorts of other pathological readouts too, and they followed what we saw with demyelination. So this could be for one of two reasons. One reason could be that the B cells that were here were bad, and they were doing something bad to the gray matter. Or depletion of these B cells makes room for other cell types to come in and potentially attenuate inflammation. And of course, we thought of IgA plasma cells because they share some of the same survival factors as B cells, namely BAFF. And I should mention that BAFF is made by astrocytes, so that there's a potential source of BAFF right here at the glial limitins. Valeria used immunofluorescence to look for IgA positive B cells in the CNS, and this is very preliminary data, but she did find that in anti-CD20 treated mice there was an abundance of these cells in the anti-CD20 treated mice as opposed to the control treated mice. And this is fairly early in the EAE response, not like the chronic EAE that I showed you earlier. So the fact that we could see any was really curious, and that we saw more of them in the anti-CD20 was also interesting. We have to back this up now using other readouts. And we also looked at the presence of IgA in the blood as well as other antibodies, and this was done by Zoa Fahim in the lab along with Valeria. And what she found was that the anti-CD20 treated mice didn't really have much of a change in, certainly not in IgM, and maybe a little bit of a decrease in IgG. But when you look at circulating IgA levels, they're actually very elevated in the anti-CD20 treated mice, along with an elevation in BAFF. And this elevation in BAFF has been seen before in humans. We know that there can be big blooms in BAFF after anti-CD20 treatment because the main consumer of BAFF, that is CD20 positive B cells, has been removed. And so we are it's tempting to speculate that this increase in IgA and IgA plasma cells in the CNS could be accounting for some of the efficacy of anti-CD20 in this model, but we have to directly prove that. But it is curious that in human, Kevin O'Connor, who has posted this paper on BioRx, has seen something kind of similar in that in myasthenia gravis patients treated with rituximab, you get cells that are resistant to Ritux, primarily plasma cells, as well as some memory B cells. And in the case of the memory B cells, the IgA positives seem to outnumber the IgG. And I don't know why this particular isotype is what's affected by anti-CD20, but if it is through an increase in BAFF or APRIL, it could be that this is actually directly stimulating TACI to make IgA, but we have yet to prove that. So in this part of the talk, what I've shown you is that anti-CD20 does improve EAE in this particular animal model of MS, and this efficacy seems to correlate at least with high IgA and BAFF. So in the last remaining couple of minutes, I want to share with you some more unpublished data on what we've been doing with respect to IgA in SARS-CoV-2 infected people. And this is work that I started 6 months ago in March with Dr. Anne-Claude Gingras at Mount Sinai Hospital in Toronto. We didn't even know each other before March, and all of a sudden we had each other on speed dial, and we were both looking at antibodies in COVID patients. So it's a big shout out to her lab and to Kento Abe and her group and for what they contributed. Now, we know what a textbook immune response looks like. It starts with IgM, then IgG, then IgA. But that doesn't seem to be what's happening with all immune responses, including SARS-CoV-2. And in SARS-CoV-2, something that's curious is that the IgA response actually comes up quite early, and it coincides with IgM. And both the IgM and IgA response appear to really crash in response to the virus. And there's some data, none of it's peer-reviewed yet, but there's some posted data from these groups here, including Michel Nussenzweig's groups and a group in France and others at UMass that have shown that IgA may actually account for some of the neutralizing antibodies that are made as a response to COVID-2. And what Michel Nussenzweig group showed was that if you swap out the FC domain for an IgA and you look at the comparative neutralizing capacity, that IgA dimers have way more neutralizing capacity than IgGs on a head-to-head basis. And that was also shown by the Wang group here too. So IgA actually might be really important in this infection, and you need to understand it. So Anne-Claude was in the process of setting up a large robotic assay to measure antibodies in huge numbers of people. Her assay can do 10,000 a day. And basically, she set up the assay in the same way that Florian Kremer had done at Mount Sinai in New York, looking at targeted antibodies to the spike part of the virus, as well as its receptor binding domain, which we all know now is what is used to bind to ACE2 on host cells. So antibodies to these particular antigens, as well as the NP antigen, nucleocapsid antigen. And then she looked at IgG, A, and M responses in her robotic setup. And this was thanks to some beautiful proteins that were made by the National Research Council, as well as our colleague James Reaney at U of T. And what Anne-Claude and Kento found was that over a 115-day period, the IgG response to the spike protein was incredibly stable. And so this is binning the patients into these different times post-infection. We also have some longitudinal data as well that back this up. The IgA response, on the other hand, crashes, and so does the IgM. So you see it early, and then it crashes down. The receptor binding domain directed antibody response for IgG is likewise very robust and mostly sustained. It dips a little bit, but again, the IgA and the IgM response crash. And this is was done in quite a number of patients. You can check out this data. It's coming out tomorrow on Science Immunology. It was accepted on Monday, and it's coming out tomorrow. We're definitely in a different world. Okay, so what we wanted to know was what did the immune response look like at the site of infection? So that's in the blood, but really we're encountering this virus in the nose and in the mouth. And is there an immune response that we can detect in those compartments? And we focused on the saliva. And this was work that was supervised by Olga, as well as Gary Chow in the lab, our project manager. And because these first-year students, Annie Pu, Baoletta, Isho, and Michelle Zhu, all really talented graduate students, had just started their degrees, I said, hey, everything's been interrupted. How would you like to pivot and work on COVID? And they were super game and did a great job. So saliva's tricky. Everyone's saliva is a little bit different. So each sample has to be titrated at multiple concentrations, and each sample needs its own normalization to no antigen controls. And we've also normalized to Ig levels, for example. So it's a tricky biofluid to work with. Titers to virus are always kind of low in the saliva, so sensitivity is key. But as I mentioned, this is worth looking at because there's a lot of IgA in the saliva, and it could be really important in neutralizing the virus. And so what we did was we collected saliva in these things called salivettes from COVID-19 patients that were provided both acute and convalescent. They were provided by Alison McGeer, who many Torontonians remember from the days of SARS in 2003, and she's always on TV, and Mary Ostrowski, a good friend from St. Mike's Hospital. And we compared those to unexposed controls from rural areas, particularly Grey Highlands, just north of Toronto, as well as pre-COVID-19 controls that we got from Saskatchewan. And what we found was that the antibody titers in the saliva were correlated quite nicely with those in the serum, which was really great. It means that we could potentially use saliva as an alternative biofluid. And that was the case for IgG, particularly for the receptor binding domain. We got this really nice correlation, but we also did quite well for spike. So about a 0.7 correlation factor. Now IgA correlated less well, as you can see here, but IgM wasn't too bad. And we don't know what to make of this yet, but it could be that the IgA response is occurring regionally and that this might account for the lack of correlation in the blood, but there is some correlation. So we're looking at this now in terms of how much of this IgA had secretory chain, for example. But so that tells us that, first of all, we can make a durable antibody response to this virus. And I think that there's been a lot of back and forth in the literature and in the preprints about this, but more and more papers are emerging, including those from Marion Pepper and Deepta Bhattacharya's lab and others that show that the antibody response to this virus is pretty solid, especially the IgG response to spike. And that these two biofluids, saliva and serum, correlate well together, at least for IgG. So it could be that the saliva could be used as an alternative fluid that's easy to collect, with the caveat that the titers are lower. And also the second caveat that we really need to look at more than one antigen, and Deepta Bhattacharya has shown that quite nicely. And that's something that we can't get out of these rapid kits. So these high throughput roboticized setups might be really ideal. But I'm going to leave you with one last piece of data before I sign off, because I don't understand it, and I'm hoping that people can respond to this on Twitter. And if you were really being sharp, sorry, I'll just skip this. If you're really being sharp, you'll notice that, so we've plotted here all of our titers for IgG, IgA, IgM to spike and receptor binding domain in the saliva. And the negatives look beautiful. There's very, very little signal for IgG or for IgM. But look at IgA. There are some patients that are some pre-COVID subjects that have some antibodies to the receptor binding domain and less so to spike. And yet the receptor binding domain is quite unique to SARS-CoV-2. And so we don't know what this is. And some of these subjects, and these are titratable saliva samples that have very low background, have titers that are up to 50% of what you'd see in a COVID-19 patient. And so we're very curious to know what these antibodies are. Do they have any relevance at all to the actual infection? Because these are people who've never seen the SARS-CoV-2 virus. And we'd like to know in longitudinal cohorts, which we're looking at now, whether this has any relevance to the early immune response to SARS-CoV-2. So that's it. I've acknowledged people as I went along. The only people I didn't acknowledge were Amit Baror and Alexandra Prat from McGill and Université de Montréal, who really, together, the three of us, started thinking about B cells in MS, and that really launched a lot of the research that I showed you today, and other really valued colleagues that I've mentioned as I went along. Thank you.
Jen, thank you so much. That was a spectacular talk. Yeah, thank you for sharing your past discoveries, and all this unpublished data is phenomenal. Thank you so much. Really appreciate it. Thank you. Carly, I think that
I'm going to
share the screen because
now.
yeah, we'll do the Twitter questions, right?
Yes, there we are. Yes, thank you, Jen. Thanks again, and for the beautiful talk, the clarity, and for teaching us that immune cells are so important in the gut-brain axis. So I really enjoy that. So just want to remind you all that you can ask questions to Jen via Twitter. There's a tweet already there that says, ask questions for Dr. Jen Gommerman, a seminar here. Please reply to this tweet. And do not forget to mention the hashtag #GlobalImmuno, and Jen is going to use her own Twitter account today. So thanks everybody. Thank you especially to Jen for your beautiful talk, and we look forward to seeing you next week.
Thanks. Bye everyone. Stay safe.
Bye.
Bye,
Thank you.
Jen. Thank you again. Bye everyone.